Retina, how does it work? What cells are in it? What do they do?
Hi, welcome to the third lecture about vision. Today we'll talk about the retina and what happens in the retina. So here's the structure of the retina. On the left you see the diagram. As you can see, it starts kind of with rods and cones. These are the receptors that actually catch the light. And because we have a habit of reading top to bottom, they are placed first, but remember somewhere in the back of your head that light on this diagram actually enters from the very bottom. So as you can see, here's the light arrows. So technically light enters this picture from the bottom and travels through all the neurons first, and it's important for understanding the physiology of the eye, but it's not that important for understanding the neural circuitry of the retina. So we will kind of stop talking about that and we'll start thinking about the propagation of the signal through the actual circuitry. Everything starts at the receptors, rods and cones. Here they are. These are the receptors. And then after that, we have several more layers of cells. And on diagrams of retina, on preparations of retina, if you make a cross section and either stain them in the old school way, so use some staining, this violet picture shows some staining of the retina, or you can make different cells glow in different colors, like this photo behind me. Either way, if you make cells visible one way or another, you notice that retina consists of layers, and it's very layered. It's differently layered in different species, so these two pictures almost certainly come from different species, and I'm not sure from which ones exactly. Probably it's the mouse behind me, but I'm not quite sure. Anyways, the layers may be slightly different. The rods and cones, as we discussed, may be slightly different. There may be relatively different concentration of different types of cells. The cells may have slightly different shape, but overall the layers in the retina are pretty much similar in different vertebrate species. So let's talk about that. Which layers are there? Everything starts with the layer of receptive cells. And you can see that they have cell bodies where the nucleus is and the DNA sits there. And then there is this part about which we actually haven't talked that much. The part that actually captures the light is a really interesting physiological and biochemical phenomenon, but we won't be talking about that in this course. And then goes the layer of bipolar cells. It's this one. Bipolar cells are called bipolar because they have two poles, which is a kind of curious way of naming a cell, but there is a cell body and it has something growing at one end and it has something growing at the other end. So it's not your archetypical neuron with a rich dendritic tree and long axon that goes to the different part of the brain. It's more like a cell body and something on top and something at the bottom. In practice, this upper part is pretty much dendrites. It listens to rods and cones, and this downward part is pretty much the axon, in the sense that it stalks to the next layer of cells. But bipolar cells are so short that they don't even bother generating action potentials. They use graded potentials. They just depolarize a bit more or hyperpolarize a bit more. And that's how they communicate, because they are short and they can't change the voltage of the entire cell. And there is no point in sending this wave of excitation, a spike, down the process that they have. Next layer is a layer where the retinal ganglion cells sit. And this is a weird name, but these cells are called retinal ganglion cells. Here in this diagram, they're just called ganglion cells. And as we discussed, a ganglion is typically a bunch of neural cells sitting somewhere outside of the brain. And this is a retina in the eye, so it's not a ganglion, not at all. The name retinal ganglion cell is kind of not helpful. It's an obsolete, old, archaic name that doesn't help anymore. And also it's very long. Retinal ganglion cells. You would get really tired if you had to use it every time you talk about the retina. So in practice, people who study this topic, they go for an abbreviation. Retinal ganglion cell becomes an RGC, and that's what people say. Just a case of a somewhat unfortunate naming, I guess. And retinal ganglion cells are important because, as you can see, these are the cells that talk to the brain. These cells have axons that are long and they collect into nerves and go from the eye into the depth of your brain. These are three main layers: receptors, bipolar cells, and retinal ganglion cells. Within these layers there are sublayers because these are not the only types of cells that are there. We also have cells that kind of sit in the middle, kind of within the bipolar cell layer, and they are kind of horizontal. So one of those is called horizontal. As you can see, it doesn't grow down, it doesn't grow up, it spreads within the layer and connects to everything around. So this type is called horizontal, and they are sitting in the bipolar layer facing rods and cones. But then there is another type called amacrine, and they are also kind of just spread within the layer. I'm trying to show one of those to you. But they are facing retinal ganglion cells. And immediately from the shape of both horizontal and amacrine cells, you can kind of intuit what they're probably doing, because see, they are reaching to the sides, which kind of implies that they're probably listening to what's happening nearby and doing something with it, right? Which kind of makes sense. If you want to figure out what you are looking at, if you want to understand the visual scene that you see, at some point, sooner or later, you will have to compare what different cones and receptors are perceiving. And from this picture, we can figure out that this process of comparison is started already in the retina. Already in the retina, you don't just communicate pixel level information to the brain, but you start doing something with this information and calculating something interesting. And we'll talk about what these cells are calculating in this lecture. Good, so what information may retinal ganglion cells communicate to the brain? Each cone, each rod, sees essentially a pixel. These pixels, as we discussed, are of different size in different parts of our visual field, and some of them are color, some of them are not. But ultimately, because we're all familiar with computers, we can think about them as elements of a picture, something like pixels. But retinal ganglion cells clearly are not just connected to their individual cones and their individual rods. So clearly something is happening within the retina. What is happening? Let's talk about that. But before we talk about that, let's just remind ourselves about these motifs, circuit motifs that we talked about in one of the previous lectures. Remember how some ways of connecting several neurons together may be more productive than some others. We talked about feed-forward inhibition, for example, and we talked about feedback inhibition. Remember, feedback inhibition, this one, is probably the most common one in the brain. And by the way, as an aside, it's kind of ironic that so many schematics from neuroscience textbooks are shown in green and red, even though we all know that this is the most common colorblind, unfriendly color scheme. So this is from the textbook, from a really good free textbook prepared by the University of Texas, but oops, it uses green and red, I'm sorry. But feedback inhibition, negative feedback on the circuit, is perhaps the most relatable one and one of the most important one. Remember, we talked about that, how a circuit tends to have this subset of inhibitory neurons that collect the average activation of the circuit and kind of feed back to the circuit. So if the circuit is overstimulated, it gets slightly inhibited. If the circuit is understimulated, it gets disinhibited and becomes more sensitive to whatever information that comes to it. It's just a generally good idea to regulate to some degree the activity of each layer in your network so that no layer would become all active or all silent because both states don't allow for any information. If everyone are shouting or if everyone are silent, both are equally unuseful. And this is probably the most relatable, but other configurations are also important. And as a preparation for our next topic, for what happens in the retina, let's do this kind of silly but helpful exercise. It's very simple, but it will, I believe, set your style of thinking into the right direction. So imagine a circuit like that. We have neurons 1, 2, 3, and 4, and 1 and 2 converge to neuron A, and 3 and 4 converge to neuron B, and then both of them converge to neuron M, and round yellowish neurons are excitatory, while square and kind of bluish neurons are inhibitory. These are the rules of the game. So let's say you can activate any two neurons in the first column. So it can be neurons 1, 2, 3, or 4. This is the only column that we can activate directly. Let's pretend that that's the case. You can activate 2 of these 4 neurons, and your goal is to maximize the spiking of this neuron. So pick any 2 from here in order to maximize spiking here. Which 2 neurons you would pick? That's assignment number 1. And then assignment number 2 is kind of the opposite. So again, you can pick any 2 neurons out of the 4 in the left, but now your goal is to minimize the spiking of the neuron M. And because minimizing is a little bit weird, like how can you minimize something that is silent? Let's assume that either they have some other inputs coming from other parts of the brain, or maybe they just spike spontaneously every now and then. So let's assume that the default is not a silent neuron, but a neuron that spikes every now and then. And in the first assignment, you need to maximize the spiking of neuron M, and in the second assignment, you need to minimize the spiking of the neuron M. Pause the video, come up with an answer, then continue and compare to my answer. It's pretty simple. Good. Let's look at that. For the first assignment, you need to maximize input in M. So let's see. It gets 2 inputs, one from the neuron A, that's excitatory, and another one from the neuron B, that's inhibitory. So to maximize M, we should maximize A, but we should minimize B. The less we inhibit M, the better. How to maximize A? One option, and in this case the only option, is to stimulate neuron 1. But how to minimize B? Let's inhibit B, and we can do that by stimulating neuron 4. Which means that for the first assignment, we need to stimulate neurons 1 and 4 in order to maximize the output of the circuit, because it would maximize excitation and at the same time minimize inhibition of the neuron M. And if you apply the same logic to the second assignment, you can end up with a different pair, because to minimize M, you want to minimize A and maximize B. So you want to stimulate B as much as possible, and you want to inhibit A as much as possible, which means that the opposite pair of neurons, 2 and 3, will be the answer for the second assignment. That should be pretty clear. And now we're prepared for discussing the retina. It turns out that if you record from retinal ganglion cells, they respond differently to different stimuli, but the most common picture is the one we see here. And let me first explain what is even the experiment here. The experiment is that we prepare a retina, either we record from a fish or some other animal where we can just connect to the retina in the whole animal, or in mice, for example, you can cut the retina out of the eye and spread it as a physiological preparation, and then you can see it with a microscope, but you can also stimulate it with light, so show some tiny pictures to it, and also record with electrodes from individual cells in the retina. And in this case, we are recording from the retinal ganglion cells. So either you're showing something to a whole eye while recording from the retina, or you spread the retina and just project something with a tiny microprojector that you can make out of a modified microscope. But either way, here's what's happening. The most common type of a retinal ganglion cell in the retina is the off cell. And here, how it works. If you record from it and everything is lit with light, it kind of spikes a little bit, but not too much. Now, if you show a dark spot in the exact middle, then you have really strong output from this type of cell. It starts spiking a lot. And this diagram below is kind of a plot of spiking. Time goes like that, some sort of voltage is recorded like that, and every time you have an action potential, it's registered as a spike. And because action potentials are narrow, it just looks as a line crossed by little changes in voltage. We talked about that before, when we talked about types of coding, for example, and how cells from one part of the brain can talk to another part of the brain, remember? So again, small dark spot in the middle, lots of spiking. Now what's interesting about these cells, if you show lots of darkness to them, instead of a small black spot, you show a large black spot, spiking happens, and we have more spiking than at rest, but less spiking than in case of a small spot. So again, to sum it up, no stimulus, some spiking, big spot, more spiking, tiny spot, lots of spiking. And they're also called off cells because they actually spike more in the beginning of the stimulus. When you just show something, they spike more, then they kind of start spiking less, but we won't be talking about this temporal dynamics right now. And in the bottom, you can see another way to stimulate this same cell. If you gradually move the area that is dark over the image, you can see that when you cross the center, you have a huge increase of spiking. And similar to the previous experiment, if everything is dark, you have some spiking, but not much. The interesting thing that's added here is that if you approach the center of the receptive field for this retinal ganglion cell, but you haven't crossed into the center yet, you have a moment when there is no spike here, when the cell goes silent. And this part, that it goes silent if you stimulate nearby but not in the center, is informative and can help us to understand what's going on. And this is how off cells work. They respond to darkness. There are also on ganglion cells that are exactly the same as off ganglion cells, except everything is reversed. They spike a little bit in the dark, they spike a little bit more at the light, and they spike the most in response to a bright spot in the middle. But because most objects that we see in the world around us are kind of dark on the white background rather than white on the dark background, if you think about that. Like most objects are more like shadows and flies and objects against the sky rather than glowing fireflies. Glowing fireflies are pretty rare. Most retinal ganglion cells in our retina are off cells, not on cells. But we have on cells as well. In this slide and in the next slide we will talk about off cells, but then you can just inverse everything and you will get a description for on cells. Just be careful about the logic and it will be fine. So basically my next question will be how you can implement something like that. But before we talk about that, watch the original video of the original experiments. The experiments of this kind were done in the 60s. And then later people recorded some documentaries about those. So there is this documentary. Watch it. It's much easier to understand what's going on if you just look how people manipulate these stimuli, and they use flashlights and all sorts of masks to project these white and dark spots on the background. All the while there is an animal looking at the screen and they record from a cell in this animal. Technically in this video they record not from the retina but from the next layer, but it doesn't change anything for the purpose of this story in particular. So pause this video, click the link in the description for on-off ganglion cells, watch it, return to this video, unpause and continue. I'll wait for you. I mean in the paused state, of course. Good, let's talk about that. Two questions. First, why it is a good thing that this is happening? This is a weird question, right? As we discussed, it's teleological, and in the first few lectures of this course, these kind of questions usually worked in the sense that most networks were optimized for something. We could infer why evolution shaped them that way. By now, you are probably, hopefully, a little bit more cynical, and you know that not all phenomena that we encounter in the brain can be explained meaningfully. Some of them are just weird. Some of them are weird and even unhelpful. But in this case, it's actually helpful, and you can figure out why this makes sense, that retinal ganglion cells, practically the first layer of processing of visual information, would perform that kind of transformation. Just think about that. Why is it useful? And then once we talk about why this is useful, think about what can be the mechanism behind it. So what happens in the retina that makes retinal ganglion cells have this complex shape of a response, provided that individual rods and cones are basically reporting pixels, right? Whether light or darkness is present here or there. Two questions. I believe you can answer them using your common sense. For the first one, just think about what animals do. For the second one, remember how we just constructed circuits from smaller cells. And pause the video, answer them, then compare your answers to mine. Let's talk about the first one. Why would you want to respond so strongly to small points, but not to larger ones? So let's think about what will happen. You will respond strongly to something like that, and you will respond much weaker to something like that. How is it helpful? If you're a frog, it can help you to catch flies. There is a different set of links in the description about how frogs catch flies, and I recommend that you watch those if you haven't yet. Indeed, that's very helpful for some animals just directly, because if you hunt for small black things and you have cells that respond to small black things, that's helpful. But why is it helpful to us? Well, if you think of it, these cells respond really weakly to a white constant color, but they also respond weakly to a dark constant color. However, they respond a lot to small points. And as we've just seen, they also respond a lot to an edge. If it's dark on one side, but bright on the other side, and the edge falls in the center of the receptive field for the cell. And you know, edges. Are edges good for vision? Are they important? Of course they are, right? Any object that you have, if you have an object, you understand where the object starts and where the object ends by looking at the edge of it. That's the first stage of vision. One of the first stages, at the very least. In order to understand what you see, you need to first understand where one thing ends and another thing starts. Is cardboard part of the wall, or is it something that stands in front of the wall? How many people are in this picture? Detecting the edge is the first step towards detecting the shape, and detecting the shape is the most important step towards understanding what object you're looking at, understanding the visual picture in general. So this kind of analysis is important. It helps you to identify points, but lines are made of points, and an edge between one color and another is a kind of a line that is also made of points. This is a good thing that the retina is doing. You can also think about it in terms of communicating changes, because as I mentioned, these cells also spike more when something appears and spike a little bit less if this dot persists for a longer period of time. This hesitancy of these cells to respond to constant color or to a prolonged presence of a stimulus is pretty similar to what we talked about before when we talked about hearing and smell, when a constant stimulus is inherently less informative. If the smell is always there, it doesn't matter. Maybe it's the smell of your own body, and there is no point in always feeling the smell of your own body. If a certain touch is always there, maybe it's just the touch of the cloth against your body, and again, there is no point in fixating on this sensation in particular. It's not useful because it's always there. So we tend to filter things that's not interesting, that's not informative, and we concentrate on things that are unusual, either in time or in space. That's what these cells are doing. So that's good. Thank you, cells. Question number 2. How can this be implemented? So let's think. We should respond, and ideally respond a lot, to activation of some rods and cones that correspond to this dark spot in the middle. But what about those rods and cones that report the presence of a stimulus around the middle? So not far away, not like really far, but immediately near the center. Okay, look. They actually don't help. Center alone, lots of spiking. Center plus surround, less spiking. And in the picture with the edge, we notice that if you only activate the surround, you not only have not a lot of spiking, you have less spiking than in the baseline state. Less spiking than for any sort of solid color, which suggests that some inhibition is involved, right? If you figured out that inhibition is involved, you did it. Good job then. So basically what happens is that the surround of the receptive field should inhibit the response. The center should cause excitation, the surround should cause inhibition. The name for that is center surround inhibition. And knowing from one of the previous slides about the structure of the retina, we know that there are some cells that stretch horizontally. There are two types, remember, horizontal and amacrine. Using common sense, we can't figure out which one of those would be responsible for that. But if you connect to them with electrodes, you can quickly figure out, okay, that's horizontal cells, not amacrine, horizontal. That's what they do. They reach out, listen to different rods and cones, and then they can create, by talking to a bipolar cell, this complex receptive field that strongly responds to activation in the middle, but is actually inhibited by activation in the area that surrounds the middle. Receptors in the center activate the bipolar cell. Receptors in the center activate the bipolar cell, while receptors around the center activate the horizontal cell that inhibits the bipolar cell, thus creating this center surround inhibition situation. So this is the case when everything is sweet and we can just reconstruct the circuit based on common sense. That's nice. Now let me spoil it a little bit. We kind of can reconstruct it from the common sense, but the reality is much weirder. And I don't expect you to remember that, so if you feel overwhelmed, just tune out. But many textbooks for first-year students kind of skip it over because they don't want people to suffer too much. But the actual situation in the retina is much weirder. It turns out, I almost feel sorry telling you about that, but it turns out that rods and cones release glutamate in the darkness. When not stimulated, they release glutamate. So all the time you lie with your eyes closed in the dark, they release glutamate. When they're stimulated with light, they stop releasing glutamate. Why it is the case, I don't know. It feels like one of those thank you evolution cases where it just doesn't make sense. Maybe it's some kind of engineering limitation. Maybe it's the only way it could have been designed, or maybe it's a case of a weird decision that happens early on and you can explain it like that. I don't know. Conceivably, if you looked at eyes of other creatures that are not vertebrates, you could get some insight about the leeway that evolution had while evolving this circuit, but I'm not that knowledgeable about other eyes, so if you're interested, you can read about that. But in the human, and in general in vertebrates, rods and cones release glutamate in the dark and stop releasing glutamate in the light, which makes no sense. But then it means that for this circuit to work, we need to now change everything slightly. The main picture doesn't change that much. There is still some gathering of information by horizontal cells, and still some inhibition is involved. But because like light is now absence of signal and darkness is the presence of signal, it turns out that for on cells, everything is turned upside down, because rods stop releasing glutamate at light, but on retinal ganglion cells are supposed to spike more in response to a bright spot. For these bipolar cells only, glutamate is inhibitory, which is also anathema, right? Glutamate is always excitatory everywhere in the brain. In all creatures, glutamate is excitatory. It's a rule that's never violated. Except as any other rule, it's sometimes violated. And in our retina, totally ignoring everything that we learned about glutamate, in some cells, not in all cells, in some cells glutamate is inhibitory. So we have something like -1 situation. Minus multiplied by minus gives you a plus. It makes no sense. I take no responsibility over it whatsoever. Thanks evolution, very helpful, very easy to remember. Fortunately, you don't have to remember that if that's an introductory course for you, but I guess if you're more serious about retinal research, you will have to deal with that. I'm sorry. And this is a nice slide about contrast sensitivity. It shows these centers around inhibition, not in a preparation, but in your own head. Let me explain to you what's going on here. So look, in your retina, you have cells that respond strongly to a tiny black spot, but respond less strongly to a larger black spot, which is kind of counterintuitive if you think about that, but yeah, we have them in the retina, lots of them, and the same for white spots, for small white spots and larger white spots. It means that potentially we would respond more strongly to a tiny object than to a larger object. It may be easier for us to see tiny objects than larger objects. Again, it sounds weird, but look at this slide. In this slide, we have different changes in tone. Some parts of it are lighter, some parts of it are darker, and the tone changes kind of oscillates like a wave. Except on the very left, these oscillations are slow, but the further you go towards the right, the faster they become. So that's the x-axis essentially. The rate, the spatial rate, the rate in space at which the stimulus changes its lightness. And bottom to up is the difference in contrast. The higher, the lower is the contrast, the lower, the stronger is the contrast, the higher is the contrast, right? And you can see, you can just look at that and figure out which part of the image you can see better and which parts are not that visible. If you're watching this lecture on a tiny phone, I'm not sure how well will it work. Maybe it actually will, maybe it won't. But at least on a screen, if you're sitting across from a normal computer screen, you may notice something interesting. And just believe me, technically, if you study this image in Photoshop or something, the difference between lighter and darker parts of this image, if you look at it at any given height, like within this horizontal line, for example, the difference is the same. But if you're like me, and you're probably kind of like me, then you can probably notice some changes in color around here, but you probably cannot notice them over here and also over here. Is it true? I hope it is true. I will pretend that it is true. And that's interesting. For these changes, it's kind of logical in the sense that the waves of darkness and lightness are so close to each other that you can imagine, okay, it's hard to see them. So when they're close to each other and the contrast is low, we don't see them at all. That's fine. But the really interesting part is that this part is often invisible. It depends on the size of your screen and on the tuning, of course. But for most people, it looks like the edge of the bars is kind of high in the middle and kind of lower on the sides. I'll stop drawing on the screen and let you enjoy the view. Because when something is rather large and blurry, the center surround inhibition kicks in and the retina just starts filtering it out. Before it even reaches the brain, it gets filtered out. So slower, spatially slower, not in time but in space, kind of more gradual changes in darkness may become almost invisible to us. Now, this is an artificial stimulus, so you don't notice it with normal stimuli, but with a stimulus like that you can notice it. And usually that's helpful, as we discussed. Usually if there is a slow shadow, kind of a very fine gradient of light from the left to the right, you don't care. If you're reading a book and the top of your book is slightly better lit than the bottom of your book, you don't notice that. When you look at the world with your own eyes, you kind of don't pay attention to these slow changes in tone. There is one object, one natural object though, that we cannot see because of that, and that's weird. And that's the closest galaxy to our galaxy. So we're totally switching gears to astronomy for a few slides, for 2 slides. Look, we have a galaxy nearby called Andromeda Galaxy. You might have heard about it. It's huge. It's huge on our sky because it's the closest galaxy to ours, and it's pretty bright. But I bet you've never seen it when you're outside, which is bizarre. It is bright, but also it has this drop of brightness towards its edges that makes the blur of this galaxy just subtle enough for our eyes, for the tiny little retinal ganglion cells inside our retina to filter it out. If it were smaller, we would have seen it as one of the brightest objects in the sky. But it is, in fact, about 6 moon diameters in width, so it's pretty huge. This picture that you see is a true representation of its size, just with an exaggerated brightness. It's very large, and this is the curse of it. Basically, here's what happens. It's brighter than the Milky Way, but you can't see the Milky Way because it's larger. You can see stars because they're smaller, but the Andromeda Galaxy has the blurry edge that's just blurry enough to hide it from our vision, which is unfortunate. In the corner, you can see a more realistic representation of just how blurry it is, and maybe from that you can intuit why it becomes invisible to a human. What a pity. Next topic, directional selectivity. Completely different topic. We're done with on-off cells for a while. Directional selectivity is not about representing the image anymore. It's about representing the motion in the image. These are cells that respond to motion. It's a different type of retinal ganglion cells. So imagine that you have this retina and you connect to a retinal ganglion cell. It's like off cell, and you connect to another one. Okay, also off cell, another one on cell. And then you connect to another one and you realize that it's not an off cell, it's not an on. The way it responds is different. And then you start showing different things to this cell, different static images and then different moving images, and you realize that this one responds to a movement, but it responds to a movement only in one particular direction and only along one particular axis. So if you have an object and this object moves left to right, you have lots of spiking. If it moves right to left, you have no spiking. If it moves top down or bottom up, you also have no spiking. There is one preferred direction and it responds to movement in this direction. These cells do really exist. We really use them to detect motion. When they are damaged, we cannot see motion. Some chemicals make them particularly active and then we see motion everywhere. But we rely on them a lot because overall our visual system is pretty slow and we have special pathways, as we'll discuss later, to process motion and understand what's moving and what's not. So in this case, it's pretty clear why these cells are useful. Let's talk about how they implement it. If you had to redesign the circuit that would support this kind of cell, how would you design this circuit? You can do that. It's a little bit harder than our previous task because now time is involved. It's not a static anymore, it's time. But imagine, so you have an object and it moves in one direction and somehow it should make one particular retinal ganglion cell spike a lot. And when the same object moves in the other direction, crossing same rods and cones, just in a different sequence, the retinal ganglion cell should be silent. Think about how you would design a circuit like that if you had to. Pause the video, think about it, then unpause and compare your answer to mine. Let's see, directional selectivity processing. What would you do? You go in one direction, like A, and then B, lots of spiking. You do B and then A, no spiking. Okay, how do we make something not spike? Inhibition. Alright, can we somehow plug inhibition in there so that AB would not harm the output, but BA would in time? We just need to compare activation of two neighboring visual receptors, rods and cones, and then we probably need to make inhibition asymmetric, right? Some sort of asymmetric inhibition feels like it should work here, so that A, B would not inhibit responses to A, but BA would. So inhibition should go in one direction but not in the other, something like that. Let's see. First of all, we need to compare activation of neighboring cells. So we'll have to deal with something like a horizontal cell or an amacrine cell. It turns out that in this case, amacrine cells are responsible. Horizontal cells are for those on-off retinal ganglion cells, while for creating motion sensitivity, amacrine cells are used, which are kind of similar, just sitting in the different layer and talking to different cells. Here how they look like if you look along the retina. So in this case, it's a real photograph and the amacrine cells are glowing. But the prettiest picture is if you look at them from above, because then you can see that they really have this really pretty shape. They have a cell body in the middle, and then they have lots of processes that talk to all the rods and cones around. They don't have axons. They don't talk to some faraway nucleus in the brain. All of these processes are a little bit like dendrites and maybe a little bit like axons as well. All of them are used to collect information, but also to some degree to send information, but mostly to collect, I guess. And we need some asymmetric inhibition. If you imagine that you have inhibition from amacrine cells, like these guys, always acting in one direction but not in the other, that would work. Because if the stimulus moves in the preferred direction, it would activate this cell and then it would activate the next cell. Once the next cell is activated, it would try to inhibit the previous cell, but it's too late. The previous cell had already spiked. While if you go in the opposite direction, in the anti-preferred direction, in the worst possible direction, you activate a cell, it inhibits the next cell, then the next cell is activated. And this inhibition that comes from rods and cones nearby manages to cancel spiking of this cell once its own rods and cones are activated. Does it make sense? You kind of have inhibition that runs a little bit ahead of you, that creates a wave of cancellations ahead of you. And thus, you can create a retinal ganglion cell that's directionally sensitive to motion. That was the hypothesis. People studied the retina, figured that out. It indeed what's happening. If you look at amacrine cells and you figure out how they connect to retinal ganglion cells, or rather they connect to bipolar cells that talk to retinal ganglion cells, but if you look at their synapses, you realize that they have synapses on one half of their beautiful dendritic body. So they collect information from this entire region, but only inhibit stuff on one side. And that creates motion selectivity, directional selectivity. And there are lots of interesting questions here. How they develop? How do they know which part is on the left, which part is on the right? We won't talk about that much, but one thing I want to point your attention at. There are 4 different types of directionally selective cells. Those that are selective to motion to the left, those to the right, those that go upwards and those that go downwards. In terms of the retina, it's called temporonasal. It's like from the temple to the nasal. Nasotemporal, and I guess these are up and down. 4 types. It's like X and Y coordinates on your screen. And that's a little bit weird if you think of that for the first time, because that's a live body, right? It's all squishy and weird and squiggly. Why would it have this system of coordinates like from your 7th grade algebra, or I don't remember in which grade people start making plots, but it looks like an XY plot. Why would you have something in your retina that looks like an XY plot? Isn't it like a totally artificial thing that humans created that doesn't exist in nature? What exists in nature? Grass and squirrels and oak trees and stones and rivers, but not XY plots, right? Well, it turns out that in the retina you have pretty much XY coordinates. They may be not exactly like that, they may be a little bit inclined one direction or another, depending on what species you are, but still it's 2 axes that are orthogonal, perpendicular to each other. And that's a little bit weird. But then if you think of that, you can kind of figure out why that's the case, because, you know, what's the prettiness of XY coordinates is that any other vector, any other direction in this system can be represented as a little bit of X and a little bit of Y. 1 axis wouldn't be enough, but 2 axes is enough. And if they're perpendicular, it makes the representation efficient in a certain way. It's just easier to work with them with 2 axes when they're perpendicular. And that's what happens in the eye. So in some ways, well, I'm getting a little bit preachy here, but I think it's interesting to reflect on the role and place of math in the world, in the way that evolution and biology reveals it. It turns out that if you want, if you need, if it just so happens that you optimize the retina, if there is a process like evolution that creates vision, and there is a retina, and there is a benefit in detecting motion, then you end up with a coordinate system. Not for pixels, but for detecting motion. It's kind of interesting. There is something to think about here. Next, I will just mention that. I won't spend too much time on that, but we haven't talked about color yet. There was motion, there was darkness and lightness. What about color? There are other retinal ganglion cells that report color to the brain. Interestingly, there are 2. One is red-green opponent cell, that's in case of humans, and another one is blue-yellow opponent cell. So they act kind of like on and off cells, except not for the darkness but for color. I guess there are several interesting parts about that. One is that even for color, there is some processing. So instead of just faithfully reporting RGB values, the retina reports the balance of opposing colors. And the colors that are chosen to be opposing are kind of weird. I don't quite understand why that's the case, but that's what we have in the retina. And the second point, the pathway for color is different from the pathway for tone, for darkness and lightness. Darkness and lightness responded to all colors, kind of, some of them more strongly than the others, but it did not deal with color. It ignored color and only looked at darkness and lightness. And that's how you detected points. While this one doesn't care about the tone. It cares about the presence or absence of particular colors in the middle and around the middle. They don't duplicate each other. They really create 2 different channels. And one thing that we'll talk about, and there will be a short demonstration for that, kind of almost like a visual illusion, we actually don't use color, as it turns out, in order to process shapes. We work with tone. We kind of see the world in black and white first, and then this color information is sprinkled on top, is added to our perception of the world, and whatever objects that we manage to identify from the black and white image getting colored. It probably sounds very abstract at this point, but we'll return to this. For now, just remember that, be aware that color is communicated with special retinal ganglion cells. And it means that by now we talked about, what is it, like 10 retinal ganglion cells. We have on and we have off, and we have directionally selective, but there are 2 axes, and for each we have left and right. So we have left and right and up and down. It's 6. And then 2 color opponents. Okay, 8, not 10. So in this lecture, we discussed 8 types of retinal ganglion cells. in general, it looks like there are about 50 distinct types of retinal ganglion cells. This is something that will be different in a mouse and in a frog and in a human. Some of these some of these 8 just have subtypes, and then there are cells that we just didn't talk about. And you can classify them by showing different stimuli and see how they respond. You also can look at the morphology, and they all will have slightly different shape, and the cell bodies will be slightly higher or lower in the layer, and they will express slightly different genes. You know, the basic story that we haven't talked about actually in this lecture so far, but when you try to understand how many different cells are in the brain, they probably have different morphology, and because they are different, they probably have slightly different channels, which means that they have slightly different genes, because after all they connect slightly differently and perform slightly different calculations, so there have to be some differences between them. The take-home message is that there are a lot, but not a ridiculous a lot, but still a lot. If you study retina, you probably would know them, but for the purposes of this class, like 8 is enough. That's all for now. See you in the next lecture.
Let us